So welcome to the late night. Gotta get this out quick because it's already almost bedtime. So, excuse me, here I want to go over beta endorphin and how there was a different coronavirus uh, in a mouse model where beta endorphin has played a role in how the pathology has either progressed or regressed. So in this particular paper, beta endorphin is being used at the time of inoculation with the virus or with the coronavirus to help lower the pathogenicity and the long-term health effects of being infected with this particular coronavirus. Now, granted, they are going to, in the discussion, talk about how if you add beta endorphin too early, you can actually increase the likelihood um, of dying or getting more serious symptoms for a different coronavirus strain. But here, I just want to discuss the sort of the importance of how these peptide hormones are playing a role in uh, affecting the pathology and the pathogenesis of coronaviruses in humans or in mammals in general. So, and also broadly, I'd like to say that this doesn't mean that we should just be giving endorphin to people to aid them against the COVID-19, because as this paper is going to demonstrate, that timing is everything in regards to when you are giving these drugs to people. Because remember, your body will oftentimes compensate for whatever drugs you're giving it. So if you give a drug and then your body ends up doing or having a response to undo the effects of that drug, that could actually make the pathology of a disease even worse. So anyways, I'm going to try to get through this quickly within the next 30 minutes. So wish me luck. So anyways, summary. The neurotropic murine coronavirus, or the MHV JHM, also known as JHMV, causes encephalitis and paralytic demyelinating disease in susceptible strains of mice and rats. So demyelinating. So remember, your nerve cells are long, and in the long part of the neurons, you have a protein that is coating the long parts of the nerve called myelin. So a demyelinating event is when that myelin gets attacked in some way, shape, or form, and then the myelin ends up degrading. So continuing in susceptible rats and serving as a model for human demyelinating diseases such as multiple sclerosis. So this is an example of a disease that results from the demyelination of your neurons. So multiple sclerosis, extremely serious, and hopefully by understanding how these demyelinating events take place, we can understand how to treat diseases such as multiple sclerosis. In this communication, we report that a single intracerebral administration, so into the brain, of naturally occurring neuropeptide beta endorphin reduces the incidence of JHMV-induced paralytic demyelinating disease by 40 to 50% in this particular strain of mice. Protection from disease was accompanied by significantly reduced virus replication in the brain as early as 3 days post-infection, and did not occur in irradiated or immunocompetent mice. So by doing this experiment on both the normal strain of the mice and on the immunocompromised versions of the mice, and sometimes you can induce immunoincompetence in creatures by giving them high dosage of radiation, and that's why they say irradiated. When you do this, the effects of beta endorphin are no longer protective. So whatever what would the word be, mechanism that beta endorphin is using to protect the mice from this JHMV virus probably works with the immune system in one way, shape, or form. Granted, there could be other cells that are killed in the process of irradiation or being immunocompetent, but for the most part, it's probably dealing with the immune system in one way, shape, or form. Anyways, the data suggests that beta endorphin engages immune mechanisms of host resistance to GHMV infection to protect the mice from disease. Introduction. The neurotropic strain of mouse hepatitis virus known as MHV4-JHM or JHMV, which will be referred to in the rest of the paper, causes acute encephalitis accompanied by acute and in some cases chronic paralytic demyelinating disease in susceptible strains of mice and rats. And it has been used extensively for the investigation of mechanisms responsible for virus-induced demyelination, serving as a model for human demyelinating diseases such as multiple sclerosis. For JHMV-induced disease, there is evidence to indicate that both oligodendrocyte death and or immune-mediated damage to myelin sheaths contribute to demyelinating lesions, depending on the viral strain and the age and genetic background of the mouse or rat hosts. The study of MHV-induced neurological disease has been facilitated by the use of variant or mutant virus strains in which specific genetic changes are associated with altered encephalotogenic or demyelinating potential. One of the GHMV mutant viruses that has been studied in detail is the neutralization-resistant strain 2.2V/1, which was selected using a monoclonal antibody specific for the spike glycoprotein of GHMV. The clinical and histopathological course of 2.2V1 infection during the first 2 weeks following inoculation has been extensively characterized in our laboratory. Briefly, viruses grow rapidly and is first detected on day 2 or 3 post-infection, post-inoculation, reaching a peak between days 3 and 5. And keep in mind of these times. So the virus is first detected on day 2 to 3 and is peaking between 3 and 5. So that is when you're getting your peak viral load. But that is in contrast when you're getting these serious chronic diseases as far as the demyelinating events or the neurological events or pathologies that the mouse is experiencing later. Anyways, a significant drop in infectious virus titer occurs on day 7, continuing to decline to undetectable levels by day 10 to 12 post-inoculation. The clinical signs of 2.2V1 infection are first apparent between days 6 to 8. So the virus is detectable on day 2 and reaches its peak between days 3 and 5. But the clinical signs are first apparent between days 6 and 8. So what can this tell you? That can tell you that a lot of the symptomology that you're experiencing is not directly from the virus itself, although there could be a delay between when the virus damages your tissues and when you first express symptoms. But what this tells me, what this indicates to me, is a lot of the problems that the mouse is experiencing from the coronavirus that it's been infected with is not from the coronavirus itself. It's how the immune system is responding to the particular coronavirus. Or in other words, the coronavirus is abusing the immune response of its host and consists of mild hind limb paralysis, which usually becomes more severe until days 12 to 14, when the disease either stabilizes or recovery begins. Demyelinating lesions are detected in the brain and spinal cord no earlier than day 6, while a characteristic infiltration of mononuclear cells first occurs on day 3 or 4 and peaks between days 6 to 9. The timing of the mononuclear cell infiltration with the drop in virus titers on day 7 post-inoculation suggests that the virus is cleared from the central nervous system by an immune-mediated mechanism. So if I remember correctly, the mononuclear cells are a form of immune cells. Continuing. In fact, a critical role for the immune response and virus clearance and protection from death has been demonstrated in previous studies using several additional JHMV strains. So there have been plenty of studies on this particular virus, just with different strains of the virus. However, in 2.2v1 infection, there's evidence that subsequent demyelination is due to immune or T cell activity in the central nervous system. So the central nervous system is basically the immune system is going into the central nervous system and then causing problems after the fact, or after the virus is in there. A similar dual role for the immune response has been reported for CNS disease, including the JHMV in the rat, and Tyler's murine encephalomyelitis virus, or TMEV, in mice. In all of these models, the immune response appears to play an initially protective role in the acute stage. So keep in mind, in all of these models, the immune response appears to play an initially protective role. So the immune response is doing what it's supposed to, and it's helping stop the progression of the virus in the acute stage, so in the short-term stage of infection, but it becomes destructive in the latter or chronic stages. So the immune response is absolutely vital and does its job in the beginning, but then if it keeps on going, if it's become dysregulated in any way, shape, or form, or if it's just a part of the natural progress of the immune system and it doesn't get a cue to stop, that is going to be causing the damage later on. In this communication, we have addressed the possibility that the naturally occurring immunomodulatory opioid peptide beta endorphin, so keep in mind, this is a peptide which is from a protein called the POMC. So the POMC is a protein that has a lot of peptides in it, and then proteases come along and separate the peptides from the protein. And these are all signaling peptides that are used for as hormones. Beta endorphin is one of them, and corticotropin is also a part of this POMC protein that gets broken up. And we do know for an absolute fact that influenza and coronaviruses are able to mimic corticotropin, at least to some degree, in the body. And by mimicking corticotropin, when your body ends up producing antibodies against the virus, it ends up attacking your own body's corticotropin. So beta endorphin, oh, and I should also mention that corticotropin is immunomodulatory. It has an effect on your cortisol levels, which in effect has an effect on your inflammatory response. So ACTH or corticotropin does that, and we do know that coronaviruses can mimic that particular peptide of the POMC protein. And then, so would it not also make sense that beta endorphin, which is a part of the same freaking protein, which is also immunomodulatory, be affected, or at least have the likelihood of being affected by these viruses as well, or related viruses? I say yes, and I think that that should be a study, or there should be focus on the opioid receptors and the opiates and the endorphins, but continuing on, we're going to keep on going with this particular paper. Anyways, it can alter the course of 2.2v1-induced paralytic demyelinating disease. The original rationale for these studies was based on the findings that beta endorphin enhances mitogen and antigen-stimulated murine T cell activities in vitro. And by reports that beta endorphin dramatically increases the severity of central nervous system disease in Swiss outbred and BALB mice, which I believe is just a strain of mouse, but I'm sure someone could go more into detail with that, infected with the temperature-sensitive mutant of vesicular stomatitis virus, or VSV, in a manner dependent on an intact immune response. The dual role that the immune response plays in the 2.2v1-induced CNS disease suggests that beta endorphin administration would either protect mice or exacerbate the disease. So this is why it is so important to do clinical trials of anything that we give to people, because sometimes if we give people a drug, it might benefit in one disease and do the exact opposite in a different disease, which is, but what we do know, or what we can easily discover, is if it does one thing or the other, right? So if it's playing a role, then we can decide whether or not we should upregulate that role or downregulate that role in order to attack the disease progression. But this is why it is so important to do clinical trials on whatever drug you're testing, because the drug might absolutely do what you want it to do in a test tube. It might do absolutely what it wants to do in a very closely related disease, but it might do the exact opposite for a disease that is currently a pandemic. So just keep that in mind. One drug or one mode of action could actually be flipped around in a closely related protein or disease. Continuing, in fact, our studies show that a single what is that? IC injection of beta endorphin, so intercerebral injection of beta endorphin at the time of infection protects these mice from both clinical and histopathological disease, accompanied by significantly reduced titers of infectious virus in the brains of treated mice as early as day 3 post inoculation. Additional data suggest that the protection is due to beta endorphin facilitated control of early virus replication that requires an intact host immune response. So what they're saying here is that additional data suggests that the protection is due to a beta endorphin facilitated control, but this beta endorphin facilitated control utilizes the intact host immune response. So it's something specifically dealing with the immune system. So sometimes you can have treatments that are beneficial not using the immune system. They could be beneficial because they are attacking the virus directly. They're attacking the cells that are infected by the virus, or they are inhibiting certain aspects of the virus in of itself. But beta endorphin seems to be acting upon whatever regulatory mechanisms are within the immune system explicitly. So materials and methods. Should I go over this? I think I will skip some of these. So basically, they have mice, and these are the particular mice that they're using, the C57BL mice. The virus preparation, so they basically cultured these viruses on mice astrocytes, which are just one of the types of brain cells. So they're culturing the virus on a monolayer of those brain cells. And then when they do the injections, that's pretty much all you really need to know about that. And then they do injections of the virus, given a 10,000 or 1,000 PFUs, which I believe is a unit of the number of viruses that they're putting into the brain. And then I think the clinical evaluation is important. All animals were monitored daily for clinical signs of virus infection in blinded fashion. So they didn't know which mice were infected or not, and they also didn't know which mice were receiving the treatment or not. So that would be one way to remove the biases of the researchers, by doing a blind study, not knowing which mice ended up receiving your beta endorphin treatment. That could remove some of the bias, because if a researcher knew which mice ended up receiving treatment, they might keep a closer eye on that particular mouse, which could end up protecting the mouse in some way, as if the researcher was paying closer attention to clinical symptoms, they might, touch the mouse more or whatever. So one way to remove that form of bias is to not even know which mice end up having the beta endorphin or not. Continuing, and they were assigned a clinical score as reported by Wang et al. According to the following criteria. So they basically rank how severe the disease progression was for these mice, and that's a way to quantify this severity of the disease. And observations continued until sacrifice at day 12 or 13 post-inoculation, when blood was obtained for serological confirmation of the virus infection by enzyme-linked immunosorbent assays. So they collect the blood to do the antibody study to make sure whether or not the mouse actually received or created antibodies. And that was done when the mouse was killed. And spinal cords were removed for histological studies. Good to know. So routine histology and immunoperoxidase staining. So just the histological studies are when they add a, if I remember correctly, they add a antibody that is targeting specific proteins so that they can do staining of those particular proteins in a microscope. So basically, if you have I just need to make sure I'm saying the right thing. Evident immunoperoxidase procedure in which the primary antibody was monoclonal. Yeah. So here they are using an antibody for the nucleocapsid protein, which then binds to that virus, and then by using that antibody as a marker to stain, you can then see where the virus was located at when you do a microscope. Because if you could imagine, if you have a sample of your tissue that you want to look at, it's very difficult to see where the viruses are at, right? Because viruses are not just microscopic, they're absolutely tiny. So to see the virus, oftentimes you need to use histological methods or antibodies to bind to those proteins of the virus, and then those antibodies can have, methods of staining so that you can see it in the actual microscope slide. So continuing. Yeah, and they're just saying that there are different ways that they could do the staining. So the results. So the intracerebral administration of beta endorphin protects mice from clinical disease induced by JHMV. So to determine the effects of beta endorphin on clinical disease induced in the 2.2 V1 5-week-old male mice received a single intracerebral injection of 1,000 PFU of virus diluted in PBS, which is a general protein solution, in the presence or absence of 1 microgram of beta endorphin. Mice, so some of them receive the beta endorphin with the initial inoculation, some of them do not. So mice were observed daily for signs of neurological disease until 13 days post-infection. The data, summarized for 8 individual experiments in Table 1, shows that overall incidence of paralysis is reduced approximately 50% in the beta endorphin-treated mice. Thus, only 16 out of 37 mice injected with beta endorphin exhibited any signs of clinical disease in 2.2 V1 infected mice. So here you can see in the table, you have groups 1, 2, and 3, and the groups had 3, 32, or 37 mice, and when they were injected, they were either injected with just beta endorphin, and as you can see here, 0 out of 3 got clinical disease when they were just injected with beta endorphin. Group number 2, so 32 of them received just the virus, and from the histological studies, they could determine that 30 out of 32 had demyelination and 30 out of 32 had clinical disease. I presume that those 30 were the same 30 here and here, but Um, Continuing, and then the third group had both beta endorphin and virus injected into their brains. And out of that group, only 16 out of 37 ended up showing clinical disease, and only 16 out of 37 had the demyelination of their brain tissues. And so, as you can see here, between 93.3% or 43.2%, I'd much rather be in the 43.2% group, indicating that with this particular timing of administration, where the beta endorphin is given at the exact same time of the viral inoculation, then you have a 50% about reduction in the clinical disease and the demyelination. Oh, and this I'll continue. So compared with an incidence of 30 to 30 of 32 in untreated infected mice. So virus infection was confirmed in all the mice, including those treated with beta endorphin, by the presence of JHMV-specific antibodies in serum prepared from blood obtained at sacrifice and measured by ELISA, and the data is not shown. The protective effect of beta endorphin and its relatively protease-resistant analog, D-ala-2 beta endorphin. So this is where they take the endorphin peptide and they replace one of the amino acids with alanine instead of what I would believe is glycine, because what is it? YGGF? I think it's YGGF is what endorphin starts with. So it'd be, if I'm remembering correctly, it would be replacing the second glycine, I should say the first glycine or the second amino acid, which is a glycine, and replacing that with alanine. And that is much more proteolytic, protease resistant than the original endorphin. Continuing, is more dramatically reflected in the clinical scores assigned in blinded fashion to infected mice and illustrated in table 2. So let's see if I can. So if the peptide was none, the clinical score, oh, and these are the days post-infection, I believe. So as you can see, if there's none, if there's no peptide that is administered at the time of inoculation, the clinical score goes up throughout those days. So it starts off at 1 and ends up at 3. And it's going up, it drops at day 10, but then continues back up. Or it's dropping at, well, it's going up to 3. So just keep that in mind. It's going up to 3. And then if they give the peptide beta endorphin with the D-alanine mutation at site number 2, that number stays pretty darn low throughout the entire time. And it is pretty much identical with, I mean, you could argue that it is a little bit lower, but it is pretty much identical with beta endorphin. And let's see, 2, 1, and 2, 3. What is going on here? I think I need to keep on reading. So yeah, I'll keep on reading and see if that shows any more. So for example, let me continue. For example, the paralysis score given to untreated infected mice was 3 on day 13 post-inoculation, compared with a score of 0.7 in the beta endorphin treatment groups. For the data presented in Table 2, the overall incidence of the disease in mice treated with either peptide was 55%. Thus, 9 out of 12 mice had no disease or a clinical score of 0, indicating that the remaining symptomatic mice were partially protected. So not only did less mice show symptoms, the ones that did show symptoms ended up having a much lower clinical score, or that they were protected even though they still showed symptoms. In addition, the data suggests that the course of disease is shortened by the beta endorphin treated mice reaching an early peak on day 8 or 9 post-inoculation, followed by almost complete recovery by day 13 post-inoculation. Untreated mice show an increase in paralysis severity until days 12 to 14 post-inoculation, with variable recovery over the next 2 or 3 months. That's a shame. 2 or 3 months after getting inoculated, and you are still dealing with some of the side effects of this particular coronavirus. Finally, there were no statistically significant differences in clinical scores between mice receiving beta endorphin or D-ala-2 beta endorphin in the experiment illustrated or in 2 additional experiments performed where the data is not shown. So they repeated the experiment and decided not to publish it because it said the same thing. Although the clinical scores tended to be lower following administration of the latter, thus, unless otherwise indicated, D-allo-2 beta-endorphin was used in subsequent experiments and for the sake of simplicity will be referred to as beta-endorphin throughout the remainder of this report. So they basically were using a more protease-resistant form of endorphin for the remainder of the experiment, but they argued that since it was not statistically different in this experiment, that they could use this more protease-resistant form of beta-endorphin for the following experiments, which goes to show you that there is some promiscuity with the opioid receptors or the receptors in your body to different forms of the same signaling peptide. So there could be a mutation every once in a while, but it can still function in many cases. So either as an antagonist or an agonist, those peptides can still have some sort of cross reactivity, even if they change an amino acid here and there. Overall, it is clear that beta endorphin treated mice, which do develop clinical disease, show a delayed onset of symptoms that are significantly attenuated or lowered in the abbreviated disease course or shortened disease course. So here's the histological evidence of protection from JHMV-induced demyelination following beta-endorphin treatment. So histological studies of spinal cords and brains obtained at sacrifice on day 13 post-inoculation revealed that the reduced incidence and severity of clinical disease is reflected in the histopathological features of the demyelination. So figure 1. So this is figure 1. So here, this column is where they receive the inoculation, and let me just double check to make sure. Yeah, and then here, B, D, and F, they received the inoculation, but they also received the beta endorphin or the beta endorphin analog. And I can't look at these and see the difference as much. Well, I can see a difference, but I don't know which one is supposed to be more disease-ridden versus the other. Granted, if I was more into this field, I probably would. But they're arguing here that for A, the Luxol fast blue stain shows demyelination in a longitudinal section of spinal cord in an untreated mouse. So longitudinal, so that would be, yeah, so spinal cord cutting through it like this. So here, apparently, you can see the demyelination versus here, which I guess this is splotchier, so they can make that claim. I'm not an expert on this, so I can't tell you which one is supposed to be disease-ridden or not. And then the inset illustrates the lesion at lower magnification at 40 times. And then B, Luxol fast blue stain on of a longitudinal section of spinal cord in the beta endorphin treated mouse showing no signs of clinical disease and no histological evidence of demyelination. So that is this one. So, I mean, I guess like you could argue since this has more, what would it be called? There's more definition between the lines. So you could argue that this one has less disease than this one, where things are much splotchier and it doesn't seem to have very good delineation between the white and the light and dark spots. I'm no expert, so don't quote me. And then continuing, immunoperoxidase staining of JHMV antigen in the white matter of the spinal cord from the same mouse as in A, and D, the JHMV antigen in the spinal cord of the beta endorphin treated mouse. Note the reduced number of antigen positive cells relative to C. So I guess here you can say that this is dealing with the myelination of the nerve cells, but this one was dealing with the sheer number of the virus particles. And here they're arguing, granted you would need multiple photos to argue whether or not there was more or less in one or the other, but for letter C, there's more dark splotches than in letter D. So you could argue that letter D, this mouse was more protected from the virus because it was able to prevent the virus from duplicating as much. And then E and F. So an E is the cross section of immunoperoxidase staining of the T200 positive cells infiltrating the spinal cord of an untreated mouse, and the infiltrating T200 positive cells of the spinal cord of the beta endorphin treated mouse. So you could argue here as well that this particular, so the T200 plus cells, those are immune cells that are mediating the immunoresponse, which might be a part of the or is pretty convincingly a part of the pathology of the actual disease that is actually causing a lot of the problems. And for E, you can see there is more white blood cells, at least the T200 positive cells, that are in the untreated mice versus the beta endorphin treated mice, where there is less. And if you are assuming that the immune response is actually causing some of the pathology, then you would want to be in this section where there is less of that immune response going on in your spinal cord. So let's see, where were we? Lesions were usually intense and focal or relatively widespread in spinal cord white matter. By contrast, the spinal cords of beta endorphin treated mice were either showed mild focal demyelinating lesions or no lesions at all, and all animals with clinical paralysis showed histological lesions regardless of treatment status. So all animals did show, with clinical paralysis, showed histological lesions. So remember, even the beta endorphin treated mice still had, half of them still ended up showing clinical symptoms. So they're saying here that all animals with clinical paralysis showed the histological lesions regardless of the treatment status. Overall, these data suggest that protection from clinical disease, largely, though not exclusively, reflects protection from demyelination. So the immunohistochemical studies showed that virus antigen was present in spinal cords obtained at day 13 from both control and treated mice, and that was figures C and D in the figures that I showed you earlier. Mice, indicating that virus is not completely cleared from the central nervous system following the beta endorphin administration. So as we said, or as I said earlier, the beta endorphins aren't completely preventing the virus from getting in. The virus is still getting in, the virus is still multiplying. Granted, here they're arguing that there's a little bit less virus going on in the beta endorphin treated mice. But also, if you're arguing that the problems that you're experiencing aren't from the virus itself, it's from your immune response, then the actual virus numbers might not be what you care as much about. However, it was frequently observed that staining for virus antigen was more intense and more widely distributed in the spinal cords of untreated mice. So as I said earlier, the untreated ones showed more evidence in the histological staining of their spinal cords relative to those of treated mice. Infiltrating T200 bearing cells were readily detected at day 13 post-inoculation in spinal cord and brains from both treatment groups. So opioid inactive N-acylated beta endorphin does not protect mice from paralytic disease. So this is to help indicate that it's the actual function of the beta endorphin that is causing the protective effects versus some random effect of the peptide itself. So the peptide might be interacting with the virus in one way, shape, or form. So we want to deconvolute that if that is possible. So anyways, many of the biological activities of opioid peptides are dependent upon the shared N-terminal opioid core amino acid sequence, tyrosine, glycine, phenylalanine, methionine, and sometimes leucine. So this would be YGGFML. And I believe, let me get the thing up. So I think this one will be better. Okay, well, here you can see I lined the POMC, or the pro-opiomelanocortin protein, from mice, and I aligned it with the spike of the mouse coronavirus that we were talking about here. Here are the you can see the YGGF of the mouse beta endorphin. So this is the active site. So here you have the lysine and arginine residue, which is a signal for a cleavage right here. And then here is the peptide that the mouse would use. And here you can see the active form, the tyrosine, glycine, phenylalanine, methionine, and here it's threonine. But that might be leucine in other mammals. So let me compare it with the human version. So YGGF. Oh no, it's also threonine in humans. So here I lined up the mouse version of the beta endorphin containing protein with the human version of it, and I aligned those to both the spike of the mouse coronavirus and the spike of the SARS-2 coronavirus, just to see where it ended up lining. So the spike of the mouse coronavirus is not lining up with where it lines up in SARS. So SARS-2 has this phenyl FGGF site, which is very similar because phenylalanine and tyrosine are very similar chemically. So having a Y versus an F is not really that much of a change. Yeah, I don't really know what to say here. I just know that the mouse coronavirus that we're talking about here does not have YGGF, but it does have multiple YGSF, which would be pretty similar. I don't know if this is YGSF. Maybe it just says one YGSF. Regardless, it does have a similar tetrapeptide, but I don't know if it's similar enough to actually be influencing this or be mimicking this particular peptide. Just know that it is technically somewhat there in the spike protein of the murine coronavirus. Continuing, modifications of this core structure by acetylation interferes with peptide binding to opioid receptors, abolishing their analgesic activities. Similarly, in 2.2V1 infection, substitution of N-acetylated beta-endorphin for the native peptide does not result in protection from paralytic demyelinating disease. So what they're arguing here is, remember when they used the D-alanine beta-endorphin? So that was a different type of endorphin that still maintains the same opioid activity as the native endorphin. And that also worked. So even though it maintained the same activity, since it still had the same activity, it still was neuroprotective. But here they're using a closely related beta endorphin that is N-acetylated, that is not active as far as its analgesic effect or its opioid effect. And if it's not doing its normal opioid effect, that is also not allowing it to function as a neuroprotective drug or treatment for this coronavirus. These data suggest that the N-terminus and perhaps opioid receptors themselves are required for the protective effects of beta endorphin. So keep in mind, if this is actually if the protection is coming from the peptide itself, then you need the peptide, or you need something similar to the peptide to do the treatment. But if the effect is the activation of opioid receptors, not the peptide itself, then perhaps using opiates is going to do the same thing. And this is something where a clinical trial would come in and determine whether or not an opiate works, or maybe an opiate agonist or an antagonist would work or not work. And it completely depends on the time of administration as well. So this is why we have clinical trials, to determine whether or not you should be just giving a drug. You can't just take heroin willy-nilly and expect it to help you out. You need to do a clinical trial first to make sure that it's actually doing what you want it to do in the body. Continuing, Table 3 also indicates that protection was observed at single doses of beta-endorphin as low as 0.01 micrograms per mouse. So a tiny amount of beta-endorphin is able to elicit the response, or at least elicit some protection. Additional dose response studies have shown that protection was not consistently observed at concentrations of 0.001 micrograms per mouse or lower. So if you go tenfold lower, you might not have a response, or enough of a response for a mouse. So just keep that in mind, I guess. So continuing, beta-endorphin treatment is associated with reduced virus titers by day 3 post-infection. So one of the possible means by which beta-endorphin may protect mice from central nervous system disease is by an effect on JHMV growth in the central nervous system. This was tested in experiments in which clarified supernatants were prepared by the brain of beta-endorphin treated and untreated mice on day 3, 5, and 9 post-inoculation, and tested for viral activity in plaque assays. So they are let me just double check to make sure I'm understanding this. This was tested in experiments in which clarified supernatants were prepared from brains. So they basically take the brains of mice, blend it, centrifuge it, and then take the top portions of it and see if the liquid on top is able to be able to infect the susceptible cells in the plaque assays or the plaque tests. And as illustrated in Figure 2, virus growth was significantly reduced in the brains of beta-endorphin treated mice as early as day 3 post-inoculation, and remained reduced throughout the period tested. The difference between the treated and untreated mice were statistically significant on day 3 and 5 post-inoculation, and at these time points, 75% of beta-endorphin treated mice had virus titers less than 100 PFUs per gram brain tissue, while the remaining 25 showed titers of 2 to 2.5 times 10 to the times 100 PFUs per gram. The data is not shown, and the variability of individual values obtained at day 9 post-inoculation, when virus growth is declining in untreated mice, resulted in a lack of statistically significant differences between the two groups. So by the time that the immune system is really working effectively, it's very difficult to get statistical significance, because there's a lot of variability, especially when the virus is in the process of being ramped down. These data suggest that beta-endorphin treatment prevents 2.2V1 from establishing a productive infection throughout the course of the 13-day disease. So if you can even prevent the virus from getting a really good stronghold on the body, you can theoretically be able to do a lot of protection by not even needing as serious of an immune response. So just make sure it never gets a stronghold on the body, and then you will not need to have the overcompensation of the immune response to overcompensate. So yeah, so here you can see the ones that did receive the beta-endorphin, or the ones that did receive the beta-endorphins and the ones that did not receive the beta-endorphins. And on days 3, 5, and 9, the ones that received the beta-endorphins had much lower virus concentrations in their brain tissue versus the ones that did not receive any beta-endorphins. Good. Continuing, and I think here they said that this was not statistically significant on day 9, but it still looks lower. So I'd like to believe that it is lower, even though it is not statistically significant. So lack of neutralizing ability of beta endorphin in vitro. Since beta endorphin is a basic peptide, it is possible that reduction in day 3 virus titers may be due to a nonspecific peptide virus interaction that prevents infection and virus growth. So this is what I'm talking about earlier. We want to make sure that the effect is done either by the peptide itself or by the regulatory mechanisms that the peptide causes in the tissues downstream. Since it is a positively charged peptide, it could have an effect on the virus duplication just from being positively charged, possibly. Anyways, this was tested in vitro by plaque assays in which various solutions of virus were added to DBT cells, which I believe is just some sort of strain of brain cells, in 24 well plates in the presence of absence of high concentrations of beta endorphin. In addition to 2.2V1, the large plaque morphology variant JHMV-DL, the large variant of JHMV, was also tested for neutralization by peptide. Plaques were obtained at 24 hours post-inoculation, and the results are presented in Table 4. The data show that concentrations of beta endorphin 10-fold higher than that capable of protecting mice from disease does not prevent infection in vitro. Interesting. So they're saying, well, this makes sense because they're just using DBT cells, which I don't know exactly what the DBT cell. We'll look this up right now. Actually, we have the technology to do this. So it's a mouse cell line. Mouse brain tumor. Okay, so it's a cell line from mice brain tumors. So that's all I'm going to look up right now. So this is a mouse brain tumor cell, and they were still susceptible to the virus in spite of the beta endorphins. But this is just the cell itself. It is not. It's not living in the mouse. So what can this tell you? This can tell you that the brain tissue in of itself is vulnerable to the virus, regardless of the beta endorphins. And the beta endorphins are not exhibiting their protective response when it is just the brain cells that are nearby. So plaques were counted at 24 hours post-inoculation, and the results are presented in Table 4. The data showed that the concentration of beta endorphin 10-fold higher than that capable of protecting mice from disease does not prevent infection in vitro. In addition, beta endorphin had no effect on the growth of either the encephalogenic strain JHMV-DL or the less virulent paralysis-inducing 2.2V1. These data suggest that it is unlikely that beta endorphin directly affects the ability of JHMV to infect or grow in cells derived from the central nervous system, and suggests that it facilitates protection indirectly by an effect on the host resistance to virus infection. So what they're saying here is it's not the beta endorphin that is causing the effect in of itself. It's not something chemically of the beta endorphin working on the neurons themselves. What is most likely causing the effect is the beta endorphins working on a completely different cell or tissue besides the neurons. So most likely it's going to be its immunoregulatory effect on the immune cells or the white blood cells. And this validates that presumption. So here they have it, beta endorphin, negative, positive, given the two different virus strains. And it looks like there's no statistical difference between them. So here, without and with, without and with for the first experiment, doesn't really look like there's that much difference. Yeah, there really isn't that a difference at all. Uh, Anyways, beta endorphin does not protect irradiated mice from clinical disease. So since clearance of GHMV from the CNS is dependent on, oh, I should back up. So what this could also tell you is that it is possible that the virus itself is using the immune system or using different cells besides the neurons to duplicate in some way, shape, or form. And if you remove all of the other aspects, you could end up with something that is not statistically significant when you are providing a treatment. So this is why it is also important to do studies not just in vitro but in vivo or in life, because sometimes what happens in the living organism can be completely different from what you experience in the test tube or in the glass. Continuing, since clearance, this is where they do irradiated mice, or they give radiation to the mice to completely destroy their immune system. Since clearance of GHMV from the central nervous system is dependent on an intact immune system, and since beta endorphins does not appear to directly neutralize virus in vitro, it is reasonable to hypothesize that its protective effect involves the immune response. To test this possibility, mice were rendered immunocompetent by whole body gamma irradiation 24 hours prior to the GHMV infection and beta endorphin treatment. Table 5 presents data which shows that beta endorphin was not able to protect mice from the central nervous system disease if the immune system was impaired. It is again evident that beta endorphin does not directly interfere with the virus infection or replication, since irradiated mice succumbed to encephalitis in spite of the beta endorphin treatment. These data provide evidence that the immune system may be required for the protective effect of beta endorphin. So here you can see the irradiated group. So it can be negative irradiated or positively irradiated, and they can either not receive beta endorphin or receive beta endorphin. And here you can see when they were both irradiated and when they were not irradiated and did not receive beta endorphin, they 7 out of 7 lived. So they all lived, but 6 of them were diseased. When they were irradiated and did not receive beta endorphin, they all died because they didn't have an immune system to save them at all. And then also they were all diseased. And then if they were not irradiated and did receive beta endorphin, they all lived like they did for the beta endorphin negative, but they also had much less disease. So here 6 out of 7 had disease, and here 2 out of 8 had disease. So it's 86% versus 25%. So that's a net positive. But when they were irradiated and did receive beta endorphin, they all died and they were all diseased. So granted, I don't know how much stock you can put into this, because if you make something immunocompetent, and it shifts the scales so far in the dying side, you might, I don't know. I don't know how much I trust this data, but they argue that beta endorphin in of itself is not enough to even put a dent into the completely immunocompromised mice. Anyways, it is again evident that beta endorphin does not directly interfere with virus infection or replication, since irradiated mice succumb to encephalitis in spite of the beta endorphin treatment. These data provide evidence that the immune system may be required for the protective effects of beta endorphin, or maybe it's also that the beta endorphin was doing an effect, but since it didn't have an immune system, it was just going to die anyway. So even if it was benefiting, nothing happened. Like they were going to die because they didn't have an immune system. So that is, I guess, also something that needs to be considered. So discussion. In this communication, we have presented data showing that a single intracerebral administration of beta endorphin protects mice from the paralytic demyelinating disease induced by 2.2 V1 a neutralization-resistant variant of JHMV. The protection was observed as reduced overall disease incidence or markedly reduced disease severity, partial protection. This is reflected in the clinical and histological evaluations. Preliminary data suggests that an opioid receptor is involved, since substitutions of opioid inactivated and acylated beta endorphin did not protect the mice from disease. So if you give them an inactivated form of beta endorphin, it does not work. The administration of beta endorphin was associated with a significantly diminished virus growth in the brains of infected mice as early as day 3 post-inoculation. The brain virus titers remained at lower limits of detection until day 9 post-inoculation, when virus growth is decreasing in untreated mice. These data suggest that beta endorphin treated prevents the establishment of a productive infection in the central nervous system. So perhaps, maybe, the virus uses the inflammatory response of the immune system to get into the central nervous system. That could possibly be why things get worse. Granted, in the completely immunocompetent mice, they still died, so I guess you can throw that theory out the window. Continuing, the finding that beta endorphin protects mice from 2.2V1 disease differs considerably from that reported of the VSV-induced disease, in which more aggressive central nervous system disease occurs when beta endorphin was administered 24 hours prior to infection. So in this different disease, they gave the mice beta endorphin a day before they did the inoculation, and that ended up making the disease more severe. So what can you gather from that? Well, your body and the body of a lot of mammals are self-regulating. So if you end up giving a drug, sometimes your body will downregulate other things to mitigate the effects of that drug. So perhaps maybe they gave the beta endorphin 24 hours earlier, which then made the mice produce less of their own beta endorphins. We don't know, but I guess we should continue. Such a discrepancy may be explained by obvious differences in these types of central nervous system disease in the two viruses caused in different strains of mice. The timing of the pathogenic events initiated by each virus, or by differences in the role that the host response plays in their pathogenicity. Thus, the temperature-sensitive strains of VSV, designated TSG31 to KS5 VSV, produces either no clinical symptoms in normal BALB mice or a slowly progressive central nervous system disease characterized by wasting, loss of appetite, hind limb paralysis, and death in these nude mice within 30 days of inoculation. In addition, the temperature sensitivity of this virus renders it more capable of replication following decrease in core body temperature, which occur following the intracranial injection of beta endorphin. So this particular strain of virus is able to duplicate much more quickly when the body decreases its temperature. So since beta endorphin can decrease body temperature, that theoretically could be causing a very large effect on how quickly the virus duplicates. And remember, this is a completely different virus at a completely different study, which is sort of contradicting the results of this particular study. By contrast, the GHMV variant of 2.2V1 causes paralytic demyelinating disease in 70 to 80% of infected these mice by day 8 post-inoculation, and has not shown temperature sensitivity when tested for growth at 33 and 39 degrees centigrade. So, should I continue? I think this is all pretty much repeating stuff that I already said. So I think we're going to leave it at this. It's getting late. I want to go to bed soon. So what does this tell you? What this tells me is that beta endorphins and the opiate receptors play some role in the immune response and they're playing some role in the inflammatory response that can either help or hurt viruses, uh, explicitly coronaviruses, from doing damage. And since we know that there is a lot of homology between this mouse coronavirus and other human infecting coronaviruses, that we should expect similar strategies that are being employed by both viruses to influence the host's immune system and influence the host's pathogenicity of the virus. Now, what would differ between most human coronaviruses and this current pandemic inducing coronavirus from SARS-CoV-2 is that this is probably the first coronavirus that is both highly pathogenic and highly transmissible in modern history. So all the other coronaviruses, so there are 4 common cold coronaviruses. None of them in the current era are highly pathogenic. They're pathogenic, but they're not highly pathogenic. So you do not need to elicit as strong of an inflammatory response to get them out of your systems, or they do not elicit as strong of an inflammatory response when you get infected with them. And part of that is arguably that the spike proteins themselves are in a completely closed state for those 4 coronaviruses, whereas in SARS, MERS, and SARS-CoV-2, they are in a partially open state, which might allow them to inject their material into the cell much more readily. Regardless, for those 4 in the current era are not highly pathogenic, and so you do not need to make a strong response against them. For SARS and MERS, those were not nearly as transmissible as SARS-CoV-2. They weren't. They simply weren't. So, and those had significant effects on even their survivors. So even if you survived from it, there were some long-term consequences that needed to be dealt with, such as depression. I won't list them all right now. What we have right now is the first coronavirus in the modern era that is both highly pathogenic and elicits strong responses in the hosts and is highly transmissible. And what I should also say is the other 4 coronaviruses that are normal that are circulating around in humans now, they might have started off highly pathogenic when they first got introduced into humans. They might have gone through seasons where they were highly pathogenic and were able to elicit strong responses. But for the common era, they haven't been. They have not been very highly pathogenic, and so they aren't eliciting the responses that are giving people like me worry or keeping me up at night. So this is the first one that is probably going to be able to sweep over the entire human population within a few years and be highly pathogenic at the same time and eliciting these strong immune responses in all these people. And what I am worried about is maybe an immune response works, it gets rid of the virus, but do you want to live with the effects of your immune system creating antibodies against this? Because this can cause autoimmune responses that you don't necessarily want to have in your body. So I just want to bring some light on why so many people in virology and pathology and in biochemistry and biophysics, why they're so worried and why they're so concerned about a virus like this. Because it's not just the virus itself that you got to worry about, it's the long-term consequences of basically having your metabolism engineered for a short period of time. So one could always make the argument that we always are surrounded by viruses at all times, and we're always fighting them off. That is also very true. But lot of viruses, not all viruses are created equal, and some are much better at hijacking your cellular machinery than others. And what we want to do is always keep that under control. Never allow yourself to get infected with a virus that throws you completely out of whack. Because even if it doesn't kill you, it will put you out of whack for a while, and that might theoretically really hurt your future and your future ability to fight off diseases. So, for instance, here, not all of the mice that got infected with this coronavirus died. A lot of them lived, but a lot of them had to deal with paralysis. Same thing with polio. A very, very small percentage of people who got polio actually got paralyzed. A very small percentage of them, less than 1%. But it was still devastating for that fraction of a percent that did end up showing symptoms. So it's late. I need to shut off now. But if you have any questions, I'm going to leave a link to this paper, and it's an old paper, so things have definitely changed since then. But it could get you into the mind of the virus and how the viruses or these coronaviruses might be able to hijack your immune system in order to accomplish what they want to accomplish. So thanks for listening, and I will see you tomorrow.